Chapter 3 Plant Kingdom Part 2 3.2.2 Mosses. The predominant stage of the life cycle of a moss is the gametophyte, which consists of two stages. The first stage is the protonema stage, which develops directly from a spore. It is creeping, green, branched, and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema as the lateral bud. They consist of upright, slender axes bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bears the sex organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organs Antheridia and Archegonia are produced in the apex of the leafy shoots. After fertilization, the zygote develops into a sporophyte consisting of foot, seta, and capsule. The sporophyte in mosses is more elaborate than that in liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common examples of mosses are Funaria, Polytrichum, and Sphagnum. 3.3 Pteridophytes. The pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purposes and as soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plants to possess vascular tissues like xylem and phloem. You shall study more about these tissues in Chapter 6. The pteridophytes are found in cool, damp, shady places, though some may flourish in sandy soil conditions. You may recall that in bryophytes, the dominant phase in the life cycle is the gametophytic plant body. However, in pteridophytes, the main plant body is a sporophyte which is differentiated into true root, stem, and leaves. These organs possesses well differentiated vascular tissues. The leaves in pteridophyta are small microphylls as in Selaginella or large macrophylls as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called as sporophylls. In some cases, sporophylls may form distinct compact structures called as strobili or cones in case of Selaginella and Equisetum. The sporangia produce spores by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous, small but multicellular, free-living, mostly photosynthetic thalloid gametophytes called as prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and the need for water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical regions. The gametophytes bear male and female sex organs called as Antheridia and Archegonia respectively. Water is required for transfer of antherozoids. The male gametes released from the Antheridia to the mouth of Archegonium. Fusion of male gamete with the egg present in the archegonium result in the formation of zygote. Zygote therefore produces a multicellular well-differentiated sporophyte which is the dominant phase of the pteridophytes. In majority of the pteridophytes, all the spores are of similar kinds. Such plants are called as homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro means large and micro that is small, spores are known as heterospores. The megaspores and microspores germinate and give rise to female and male gametophytes respectively. The female gametophytes in these plants are retained on the parent sporophytes for variable periods. The development of the zygotes into young embryos take place within the female gametophytes. This event is a precursor to the seed habit, considered an important step in evolution. The pteridophytes are further classified into four classes: Psilopsida, Xylotum; Lycopsida, example Selaginella, Lycopodium; Sphenopsida, example Equisetum; and Pteridopsida, example Dryopteris, Pteris, and Adiantum. 3.4 Gymnosperms. The gymnosperms, gymnos means naked and sperma means seeds, are the plants in which the ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. The seeds that develop post fertilization are not covered, that is, they are naked. Gymnosperms include medium sized trees or tall trees and shrubs. One of the gymnosperms, the giant redwood tree sequoia, is one of the tallest tree species. The roots are generally tap roots. Roots in some genera have fungal association in the form of mycorrhiza, pinus, while in some others, cycas, small, specialized roots called as coralloid roots are associated with nitrogen fixing cyanobacteria. The stems are unbranched in case of cycas or branched in case of pinus or cedrus. The leaves may be simple or compound. In cycas, the pinnate leaves persist for few years. The leaves in gymnosperms are well adapted to withstand extremes of the temperature, humidity, and wind. In conifers, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also help to reduce the water loss. The gymnosperms are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within the sporangia that are borne on sporophylls, which are arranged spirally along an axis to form lacs or compact strobili or cones. The strobili bearing microsporophylls and microsporangia are called as microsporangiate or male strobili. The microspores develop into a male gametophytic generation, which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called as pollen grain. The development of pollen grains take place within the microsporangia. The cones bearing megasporophylls with the ovules or megasporangia are called as macrosporangiate or female strobili. The male or female cones or strobili may be borne on the same tree in case of pinus. However, in cycas, male cones and megasporophylls are borne on different trees. The megaspore mother cell is differentiated from one of the cells of the nucellus. The nucellus is protected by envelopes and the composite structure is called as an ovule. The ovules are borne on megasporophylls which may be clustered to form the female cones. The megaspore mother cell divides meiotically to form four megaspores. One of the megaspores enclosed within the megasporangia develops into a multicellular female gametophyte that bears two or more archegonia or female sex organs. The multicellular female gametophyte is also retained within megasporangia. Unlike bryophytes and pteridophytes, in gymnosperms, the male and female gametophytes do not have an independent free living existence. They remain within the sporangia, retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules borne on megasporophylls. The pollen tube carrying the male gametophytes grows towards archegonia in the ovules and discharge their contents near the mouth of the archegonia. Following fertilization, zygote develops into an embryo and the ovules into seeds. These seeds are not covered. 3.5 Angiosperms. Unlike the gymnosperms, where the ovules are naked, in the angiosperms of flowering plants, the pollen grains and ovules are developed in specialized structures called as flowers. In angiosperms, the seeds are enclosed in fruits. The angiosperms are an exceptionally large group of plants occurring in wide range of habitats. They range in size from the smallest Wolffia to tall trees of eucalyptus over 100 meters. They provide us with food, fodder, fuel, medicines, and several other commercially important products. They are divided into two classes, the dicotyledons and the monocotyledons. The dicotyledons are characterized by seeds having two cotyledons, reticulate venation in leaves, and tetramerous or pentamerous flowers, that is, having 4 or 5 members in each floral whorls. The monocotyledons, on the other hand, are characterized by single cotyledonous seed, parallel venation in leaves, and trimerous flowers having 3 members in each floral whorls. The male sex organ in a flower is the stamen. Each stamen consists of a slender filament with an anther at the tip. Within the anthers, the pollen mother cell divides by meiosis to produce microspores which matures into the pollen grains. The female sex organ in a flower is the pistil. Pistil consists of a swollen ovary at its base, a long slender style, and a stigma. Inside the ovary, ovules are present. Generally, each ovule has a megaspore mother cell that undergoes meiosis to form 4 haploid megaspores. 3 of them degenerate and one divide to form the embryo sac. Each embryo sac has a 3-celled egg apparatus, 1 egg cell and 2 synergids, 3 antipodal cells, and 2 polar nuclei. The polar nuclei eventually fuse to produce a diploid secondary nucleus. Pollen grain, after dispersal from the anthers, are carried by wind or various other agencies to the stigma of a pistil. This is termed as pollination. The pollen grains germinate on the stigma and the resulting pollen tubes grow through the tissues of stigma and style and reach the ovule. The pollen tubes enter the embryo sac where 2 male gametes are discharged. One of the male gametes fuses with the egg cell, that is syngamy, to form a zygote. The other male gamete fuses with the diploid secondary nucleus to produce the triploid primary endosperm nucleus, that is PEN. Because of the occurrence of two fusions, that is syngamy and triple fusion, this event is termed as double fertilization, an event unique to the angiosperms. The zygote develops into an embryo with 1 or 2 cotyledons, and the PEN develops into endosperm, which produces nourishment to the developing embryo. The synergids and antipodals degenerate after fertilization. During these events, the ovules develop into seeds and the ovaries develop into fruit. The life cycle of an angiosperm is shown in figure 3.6. 3.6 Plant life cycles and alteration of generations. In plants, both haploid and diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies, haploid and diploid. The haploid plant body produces gametes by mitosis. This plant body represents a gametophyte. Following fertilization, The zygote also divides by mitosis to produce a diploid sporophytic plant body. Haploid spores are produced by this plant body by meiosis. These in turn divide by mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alternation of generations between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals representing them differ in the following patterns. First, sporophytic generation is represented only by the one celled zygote. There are no free living sporophytes. Meiosis in the zygote results in the formation of haploid spores. The haploid spores divide mitotically and form the gametophyte. The dominant photosynthetic phase in such plants is the free living gametophyte. This kind of life cycle is termed as haplontic. Many algae, such as volvox, pyrogyra, and some species of Chlamydomonas, represent this pattern. Second, on the other extreme is the type wherein the diploid sporophyte is the dominant photosynthetic independent phase of the plant. The gametophytic phase is represented by the single to the few celled haploid gametophyte. This kind of life cycle is termed as diplontic, and Alga fucus species represent this pattern. In addition, all seed-bearing plants, that is gymnosperms and angiosperms, follow this pattern with some variations, wherein the gametophytic phase is few to multicelled. Third, bryophytes and pteridophytes interestingly exhibit an intermediate condition that is haplodiplontic. Both phases are multicellular. However, they differ in their dominant phases. A dominant, independent, photosynthetic thalloid or erect phase is represented by a haploid gametophyte, and it alternates with the short lived multicellular sporophyte, totally or partially dependent on the gametophyte for its anchorage and nutrition. All bryophytes represent this pattern. The diploid sporophyte is represented by a dominant, independent, photosynthetic vascular plant body. It alternates with multicellular, saprophytic or autotrophic, independent but short-lived haploid gametophyte. Such a pattern is known as haplodiplontic life cycle. All pteridophytes exhibit this pattern. Interestingly, while most algal genera are haplontic, some of them, such as Ectocarpus, Polysiphonia, kelps, are haplodiplontic. Fucus and Alga is diplontic. Summary: Plant kingdom includes algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. Algae are chlorophyll bearing, simple, thalloid, autotrophic, and largely aquatic organisms. Depending on the type of pigment possessed and the type of stored food, algae are classified into three classes, namely chlorophyceae, phaeophyceae, and rhodophyceae. Algae usually reproduce vegetatively by fragmentation, asexually by formation of different types of spores, and sexually by formation of gametes, which may show isogamy, anisogamy, or oogamy. Bryophytes are the plants which can live in soil but are dependent on water for sexual reproduction. The plant body is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by rhizoids. They possess root-like, leaf-like, and stem-like structures. The bryophytes are divided into liverworts and mosses. The plant body of liverworts is thalloid and dorsiventral, whereas mosses have upright, slender axes bearing spirally arranged leaves. The main plant body of a bryophyte is gamete producing and is called as gametophyte. It bears the male sex organs called as antheridia and female sex organs called as archegonia. The male and female gametes fuse to form zygote, which produces a multicellular body called as sporophyte. It produces haploid spores. The spores germinate to form the gametophytes. In pteridophytes, the main plant body is a sporophyte which is differentiated into true root, stem, and leaves. These organs possess well differentiated vascular tissues. The sporophytes bear sporangium, which produces spores. The spores germinate to form the gametophytes, which require cool, damp places to grow. The gametophytes bear male and female sex organs called as antheridia and archegonia respectively. Water is required for the transfer of male gametes to archegonium, where zygote is formed after fertilization. The zygote produces a sporophyte. The gymnosperms are the plants in which ovules are not enclosed by any ovary wall. After fertilization, the seeds remain exposed and therefore these plants are called as naked seed plants. The gymnosperms produce microspores and megaspores, which are produced in microsporangia and megasporangia borne on the sporophylls. The sporophylls, that is, microsporophylls and megasporophylls, are arranged spirally on axis to form male and female cones respectively. The pollen grain germinates and pollen tube releases the male gamete into the ovule where it fuses with the egg cell in archegonia. Following fertilization, the zygote develops into embryo and the ovules into seeds. In angiosperms, the male sex organs, that is stamen, and female sex organs, that is pistil, are born in a flower. Each stamen consists of a filament and an anther. The anther produces pollen grains, that is male gametophyte, after meiosis. The pistil consists of an ovary enclosing one to many ovules. Within the ovule is the female gametophyte or embryo sac, which contains the egg cell. The pollen tube enters the embryo sac where two male gametes are discharged. One male gamete fuses with the egg cell, that is syngamy, and other fuses with the diploid secondary nucleus, that is triple fusion. This phenomenon of two fusions is called as double fertilization and it is unique to angiosperms. The angiosperms are divided into two classes, the dicotyledons and the monocotyledons. During the life cycle of any sexually reproducing plant, there is alternation of generations between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals may show different patterns of life cycles, that is haplontic, diplontic, or intermediate.